production. In the next part, I want to tell you how we've set up the dynamic flux balance approach and how we tested it with different models. And in the last part, I want to give some insights how we can valorize the residual biomass, because the beta-carotene, our high-value product, is only a small fraction of the biomass, actually. Maybe my presentation is too large and that's why it takes so long. Okay, so the biological system, as already explained, is Dunaliella salina. It produces high fraction of beta-carotene. That's why it's an interesting production organism. From all carotenoid pigments produced in microalgae, beta-carotene has the largest market value, and it's because the algae is halotolerant, so it grows in very salty cultivation mediums. It's very interesting for outdoor cultivation of algae, and the algae has actually two different modes. The green mode, where it's normally producing biomass, and if you switch the light and nutrient conditions, the cells underwent a metabolic switch, and then they produce the carotenoids, which you also see in the right part. It's a picture taken in San Francisco, and if the cells start accumulating carotenoids, you can see it from the air that it's changing the color. So I want to show you briefly how the industrial process of beta-carotene production looks like. In the left part, you have the upstream processing part, where you cultivate and grow and the algae and produce the beta-carotene. And in the right part, you have the harvesting and downstream processing procedure, where in the end, the beta-carotene is normally extracted via solvent extraction, and it accounts up to normally 10% or even lower of the biomass, and you have more than 90% of low value residual biomass left. And that's our hypothesis that we can, first we should of course optimize a high value product, and in the end we should think about what can we do with the residual biomass. So coming back to the outline, I now want to tell you how we implemented this dynamic flux balance approach. So here we have a picture of our simplified algal cell, which can produce in the metabolism different compounds, and it's embedded in a reactor environment, and light and nutrient, and also in outdoor conditions, temperature, and a lot of things are changing, and this is accounted for in the model, what we call the extracellular effects, like the reactor dynamics, light attenuation, and nutrient supply. And from the metabolic point of view, we have the underlying metabolic network, which then leads to formation of certain products, and this is covered by this box of the intracellular effects. And now I want to first focus on the extracellular effects, how we have determined the kinetic parameters for these reactor dynamics. We have set up a very simple dynamic kinetic model, which covers for the most important biomass properties that we can describe: light attenuation and nutrient consumption in the reactor with simple ordinary differential equations. We have concentrated on biomass density, meaning how fast is the biomass growing, the chlorophyll fraction, so the photosynthetic pigment, which is important to describe the light attenuation in the reactor. And the last properties are referred to the nutrient status in the medium and inside the cell. And we have evaluated this experimentally under 3 different conditions, mainly under low light conditions, abbreviated as LL, where the cells are in this green state and they are just growing without producing a significant amount of carotenoids, and then highlight intensities and high nutrients, where the cells could accumulate beta-carotene to protect from the highlight intensities, and the combination of these highlight intensities with the low nutrients then leads to the highest beta-carotene amount in the cells. And what we can see in the next slide, this is a prediction of our state variables. The biomass density in the first line under these conditions. In the second row, the nitrogen density, chlorophyll fraction, and these intracellular nitrogen fraction, which are covered quite well from the model, which is characterized or estimated with 9 kinetic parameters. And in addition to that, we also checked if our parameters are identifiable. We used a profile likelihood approach, and you see the results of 6 parameters depicted exemplarily there for the nitrogen uptake. The dotted line is a normal sensitivity analysis, so we assume that all parameters are not dependent or not structurally related to each other. And you see that for all examples there, that the sensitivity analysis led to the smallest confidence intervals. But if we also allow for structural dependencies between the parameters, which are depicted in the black line there, we see that we get much larger confidence bounds. And if we, for example, measure one experimental data point like here, and we include this in the model, we get to these blue lines where we see that we can also, if we account for structural dependencies, if we have more information about the system, we can decrease the confidence intervals. And out of 9 parameters, we had only 2 parameters which were not identifiable, and we designed some experiments to determine the measured values for these parameters. And with this optimized set, we now go back to our dynamic flux balance approach and now have a closer look to the stoichiometric metabolic network. And because our alga Dunaliella salina is a sequence, but the genome is not annotated, and therefore we don't have a metabolic network representation, we made use of another green microalga, namely Chlamydomonas reinhardti. And we wanted to see if we can also describe, because all the essential features were covered in there, if we can describe our biomass composition and the growth. And we have the 4 state variables which we had in the previous reactor model as well. And we added storage for beta-carotene. And then we combined our extracellular dynamics with the molecular level represented by this metabolic network. And then I'll present you the results for our experimental data set. And here we were quite satisfied. The 4 state variables from the reactor model here are covered well. And in addition, we are able to describe the beta-carotene under accumulation under 3 different reaction conditions. Now I want to tell you something about the performance of this genome scale model of Chlamydomonas reinhardti in comparison to a reduced network model, and how we use this to describe the biomass composition, which we need for the biorefinery approach or for the valorization of the residual biomass. We have used as a framework reduced metabolic model, which has been developed by the group of René Weifels and Wageningen. And we added, to cover all essential features of the Dunaliella metabolism, we have added the pathways for carotenoid production to this reduced network, which consists of roughly 170 reactions. And we considered more biomass fractions than in the previous model, which are important biomass constituents like neutral lipids, protein, starch, and glycerol, which in some these 6 correspond to up to 90% of the whole biomass. Yeah, and as I already said, this facilitates then the development of an biorefinery approach for this special microalga. And here you see in the dotted lines the performance of this reduced model in comparison to the genome scale model in solid. And for the low light conditions, we are quite satisfied with the prediction results, but there are some deviations for the high light conditions where the reduced model cannot, because of this reduced structure, not get the accuracy as the genome scale model, but we are currently also working on improvement. And in the last part of my talk, I want to show you the results of these residual biomass. So here in orange, we have our beta-carotene. In yellow, we have the neutral lipids, which are stored together with the beta-carotene in globules or vesicles. And in blue here, we have a small protein fraction. It's 10% of the biomass. If you would have more protein fraction in the cell under different cultivation conditions, for instance, you could potentially use it as an animal feed, but like if you have such a small fraction, it's maybe not that valuable. Glycerol as a side product is also not very valuable in our view, but you could use it for mixotrophic algae cultivation. There are some species which grow on glycerol, but the highest fraction we observed in our analysis and also in the experiments as a large starch fraction, 50 up to 70% we measured in our biomass, and we tested an approach of hydrothermal liquefaction to make use of these large carbohydrate fraction in the cell. And this leads me to the results of my colleague Christine Hurwitz. She has extracted the beta-carotene because it's our high-value product with solvent extraction from the biomass. And these extracted residual biomass have then been subjected to hydrothermal liquefaction. And they call it mild hydrothermal liquefaction because they use reaction temperatures from 100 to 200 degrees. Normally their temperatures are above 300%. And we found in the aqueous phase, which is over 90 or over 80% of the liquefaction phase, that there are very high glucose concentrations in this aqueous phase, so around 70%, even under very mild conditions. The organic phase, which is usually considered as a biofuel phase, is very small, below 10%, because we have already solvent extracted the biomass, and so the carotenoids and neutralipids weren't available in this liquefaction. So we focus on these aqueous phase. And she tested the growth of some biotechnologically relevant organisms like Escherichia coli, Saccharomyces cerevisiae, and Chlorella vulgaris. And here you see the results. So in black, you have here the growth on control medium, and in red, gray, and green, which is most important, you have the growth on our modified media, where we have substituted the glucose in the control media by this liquefaction aqueous phase. And you see that the growth is very comparable. So we conclude that there are no toxic compounds in this liquefaction phase, and that it would be a good idea potentially to use this aqueous phase as a substrate for mixotrophic or heterotrophic processes. So with this, I hope I stay in time, and I want to summarize. Yeah, I have shown you how we have developed from a simple reactor model, this dynamic flux balance model of Dunaliella salina. And this gives us some implications how we can modify or optimize the up and downstream processing of this system. For instance, what are optimal harvesting time points, and how can we make use of the residual biomass? And as an outlook, I want to mention that we are currently working on a metabolic network representation of Dunaliella salina from genetic data that has been provided by Jürgen Poller from the Brooklyn College in New York. And with this, I thank you for your attention.
Thank you very much. We have time for questions. Please introduce yourself.
Yeah, I'm Adriano Encinas from Brazil. Nice presentation. I would like to ask about your modeling, growth modeling work. Do you include temperature as a variable?
Not so far, because we have only lab scale data so far, but we want to extend this approach also to other species, and we want to implement a cooperation with different institutes in Germany which are growing algae on large scale, and there we definitely have to include this, but there you have also so much other effects. You have contaminations and maybe mixed species, and I'm not sure if also DFBA is a valid or a good approach for that, but we are going to test this, because for Dunaliella in Germany we don't have so good cultivation options to get comparable data as it is achieved in production facilities like in Israel or in Australia.
Okay. I have a second question.
Thank you. It was very interesting. I also have two questions, so I'll just do my first question. So, as the conditions are changed to stimulate the beta carotene accumulation, how much reduction in growth rate do you get?
So,
I assume there's some.
so if from the initial cultivation phase we had some residual nutrients, and there the growth rates are comparable to other cultivation conditions, but if the nutrients get depleted, the growth rate stops almost immediately or during one day. So it doesn't grow anymore.
Okay, so but you need to do that to stimulate the beta carotene. So you grow it under nutrient-complete conditions.
Yeah.
Okay.
We have also done an approach where we have implemented an optimized fed batch process, where we try to tackle this approach where we feed it nutrients continuously in a certain relation to the biomass which was in the reactor, and compared to our standard process we had almost doubled the beta-carotene density and the biomass density. So the batch is maybe not the best condition, or you have to tune these initial nutrient concentration in your reactors in order to achieve a high biomass density before the cells run out of nutrients. Thank you.
Okay. My second question is about the extraction, the beta-carotene. Do you use a wet or dried biomass extraction?
We use
Thinking about the use of the residual thermal coefficient.
we use dried biomass. It's spray dried as far as I know, so I'm not conducting the extraction results, but my colleague is also doing experiments with supercritical solvent extraction, and there she compares also the use of dry versus wet biomass. So up to 10% of raw water remaining in the biomass, I think she is considering.
Yeah, thank you for the presentation. Very good presentation. You mentioned about German technology, whatever, but BASF is the company that produces large amount of carotene in Australia. Yeah. So my natural question is how your technology, your developments, compare with the established knowledge that they have from BASF on these plants over there.
So we have a cooperation, not with the BASF, but with Ami Ben Amos, which is operating the facility in Israel, and he provided also data for us, and this is quite comparable.
So how it compares to what it is known on Dunaliella? This is my question. Now they have the plants in Australia, which is German technology, is BASF. It's Ben Amos in Eilat, which has MBT. So how what you produce compares with what they do?
I think we can't compare it because we produce under lab scale conditions, very controlled bioreactors. So I think it wouldn't make any sense to make a cost calculation for that process. So I'm not sure how I answer your question adequately.
I have a second question, if you allow. You mentioned about unless they
I
got.
can ask.
And the second is about the reactors. Yeah. Based on the experience, including the names that you also referred to, the experience is that you cultivate in open forms. Yeah. Whereas in your experiments, you have a control reactor, right?
Yeah.
Which is more the case of the PBR.
Yeah.
And it's known that the PBR is not good for the Dunaliella because it sticks on the surface of the tubes and whatever, and it's not good.
Yeah, maybe
So
I think also.
So why not taking into consideration that you have this sort of reactor in the end, and you should be focusing on this one and not control reactor?
We used Dunaliella as a first case study because we have started these algae experiments and modeling 4 years ago roughly, and we wanted to use methods which are developed or which have been developed in our group for the chemical engineering and transfer to biological systems. So we decided first to take an organism which is well known in industry and where we also know literature parameters, and but now we want to extend this approach also to open cultivations. I think for Dunaliella, for this case, it doesn't make sense to cultivate it in closed reactors because it's also salt tolerant and it's not so prone to invaders as other freshwater microalgae, for instance. But yeah, we want to transfer this approach, but I'm not sure. We are also testing different modeling approaches. If this DFBA framework is a good framework for open pond cultivations, for instance, because we have so many other effects, and then maybe the metabolic network information is not the information which drives the accuracy of the prediction.
Okay, thank you very much.